Hello, my dear students. Welcome you all to Sri Jaindra Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, in the last lecture, we discussed about the anatomy of the urinary system that includes kidney, urinary bladder. We discussed about the histology of different parts of urinary system, that is kidneys, ureters. Okay, so today I am going to start with the physiology part, that is the physiology of urine formation, the physiology of excretion. Okay, after discussing this, the physiology of excretion, which basically involves the mechanism of urine formation, we will be discussing about the anatomical features of urinary bladder. What is the different types of muscles, walls, okay, and different types of tissues which are present in urinary bladder. Okay, the physiology of excretion is today's topic. The urine formation occurs in mainly three steps. One is the glomerular filtration. The first step of urine formation in three major steps. One is glomerular filtration. Second, selective या tubular reabsorption or tubular. And the third is tubular secretion. Okay. So the process of formation of urine is basically conducted in these three steps. One is the glomerular filtration. The second one is selective or tubular reabsorption. And the third one is tubular secretion. Now, before this, the major nitrogenous excretory substances in frog, rabbit, human is urea. I mean, we all are ureotelic organisms. These are ureotelic animals. The excretory physiology in these animals may be considered under two phases. One is the formation of urea. We have already discussed about the ornithine cycle, Krebs-Haslet cycle, and formation and excretion of urine. So protein, they are digested in different types of, with the help of different types of digestive juices in the alimentary canal to form amino acids, which further absorbs in intestine. Okay, they become the part of the blood circulation. Then the harmful amino acids, they enter in the liver and get biometabolized into liver, and the useful amino acids, they get assimilated in the cells to form the proteins, dietary proteins. Okay, so these amino acids, which are non-proteinaceous, which are, we can say, the essential amino acids and the non-essential amino acids. The essential amino acids are the one which are required for making the developmental proteins, enzymes, and the non-essential amino acids which are required but they are not required or they are not functional as a enzyme. So they basically enter in the liver and get biometabolized in the liver to form the nitrogenous waste, that is urea. Urine formation. It occurs in kidneys and involves three processes: glomerular filtration, reabsorption, and tubular secretion. Okay. Now we are going to talk about the glomerular filtration, that is ultrafiltration. Now, very interestingly, we have to study about the Starling's hypothesis. So, according to Starling, it is a passive process. It is a passive process which takes place from the glomerulus into the boundless capsule. The glomerular epithelium has various micropores, diameter 50 to 100 nanometers or 0.05 to zero point 0.1 millimeters, which increase the rate of filtration. So it is a non-selective passive process. It is a non-selective passive process which takes place from the glomerulus into the boundless capsule. The glomerular epithelium has various micropores. Size of the micropores are different. Diameter is around on an average 50 to 100 nanometers or 0.05 to zero point 0.1 millimeters, which increases the rate of filtration. Okay. There are many favorable pressures and many opposing pressures which are responsible for making the effective filtration pressure, right? The non-colloidal part of the plasma has urea, water, glucose, salts, vitamins, minerals, nitrogenous waste are forced out from the glomerular capillaries into the boundless capsule. So the non-colloidal parts such as urea, water, glucose, salts, vitamin, minerals, nitrogenous waste are forced out from the glomerular capillaries into the boundless capsule by high pressure of the blood in the glomerular capillaries. By the high pressure of blood in the glomerular capillaries. The pressure and the resistance is very high because the glomerular capillaries are narrower than the afferent renal arteries. The glomerular capillaries are about 50 times more permeable than the capillaries elsewhere. The size of the glomerular capillaries is narrow in comparison to the afferent arteriole, which is, bringing the blood into the glomeruli. The glomerular capillaries, that is why they are 50 times more permeable than the capillaries elsewhere. Pressure is highest in glomerular capillaries than in the capillaries elsewhere produce. That is why it produce more amount of filtrate. Okay, is it understood? Now we are going to talk about the process of ultrafiltration or the glomerular filtration. First I will talk about that this non-selective passive process in which the blood plasma is filtered. The blood plasma is filtered due to blood plasma due to glomerular hydrostatic pressure. Glomerular hydrostatic pressure. This is the glomerular hydrostatic pressure. Okay. Now, the pressure which is favorable filtration is glomerular hydrostatic pressure. It is mainly due to broad afferent arteriole and narrow efferent arteriole. Okay, it is around 60 to 70 millimeters of mercury. The range is around 60 to 70 millimeters of mercury. Okay, just give me a minute. Yeah, so this glomerular hydrostatic pressure is because the afferent arteriole, which is bringing the blood towards glomeruli is broad in size and it is narrow in size. So because of narrow in size, the blood it undergoes stasis. It undergoes stasis. Okay. So this stasis will lead to forcing on the non-colloidal filtrate, that is the urea, glucose, everything they will leak out. Okay. So this is the glomerular hydrostatic pressure. Okay. The effective filtration pressure that causes ultrafiltration is determined by three pressure. One is the glomerular hydrostatic pressure, which is favorable. Okay. The blood colloidal osmotic pressure, BCOP, and capsular hydrostatic pressure, they are non-opposing. Blood colloidal osmotic pressure, okay, it is due to albumin protein in blood. This is due to albumin protein in blood. Okay. And this capsular hydrostatic pressure, it is basically due to fluid in Bowman's capsule. Okay. Glomerular blood hydrostatic pressure. The hydrostatic pressure is a force that a fluid under pressure exerts against the walls of its container. So glomerular blood hydrostatic pressure, it is basically the pressure which is exerted by the fluid under pressure against the walls of its container. It is around glomerular blood hydrostatic pressure, also known as the glomerular hydrostatic pressure. It is around 70 millimeters of mercury. Then the blood colloidal osmotic pressure is the osmotic pressure created in the blood of glomerular capillaries due to the plasma protein albumin, globulin, and fibrinogen. It resists the filtration of the fluid from the capillaries. The blood colloidal osmotic pressure is basically because of the colloidal substances like proteins. So these proteins, when they are the part of stream of blood flow, so they create a resisted pressure, they create a resistance against avoiding the leakage of the materials, leakage of the non-colloidal part, that is the urea and everything. So this blood colloidal osmotic pressure is the osmotic pressure created in the blood of glomerular capillaries due to the plasma proteins like albumin, globulin, fibrinogen. It resists the filtration of the fluid from the capillaries. It is around 30 millimeters of mercury. Okay. Blood colloidal osmotic pressure in the other body capillaries is 25 millimeters of mercury. So you can see the blood colloidal osmotic pressure in these arteries or capillaries, the glomerular capillaries is 30 millimeters of mercury, while in the other body capillaries it is 25 millimeters of mercury. Capsular hydrostatic pressure. The capsular hydrostatic pressure is the pressure caused by fluid that is filtrate that reaches into the Bowman's capsule and resist filtration. It is around 20 millimeters of mercury. Okay. So the glomerular blood hydrostatic pressure or the glomerular hydrostatic pressure, then blood colloidal osmotic pressure, and the capsular hydrostatic pressure. So the minus the blood colloidal osmotic pressure plus the capsular hydrostatic pressure. It is around 70 minus 30 plus 20. That becomes 20. Okay. Or if it is 60 minus 30 plus 20, then it's 10. The range of G, the glomerular filtration rate is 10 to 20, or the pressure which is required, which is favorable to pursue the glomerular filtration, that is 10 to 20 millimeters of mercury. Okay. Now, the most important thing about GFR, the glomerular filtration rate, it remains nearly same. GFR, it remains same. as it undergoes autoregulation. Okay. Nitrous oxide, NO, is a potent vasodilator. Proteins, they are not present in glomerular filtrate due to their large size. Okay. So GFR, it remains same because it undergoes autoregulation. Nitric oxide is a potent vasodilator, and the proteins which are responsible for blood colloidal osmotic pressure, they do not pass through the slits because their size is high. The effective filtration pressure, effective or the net filtration pressure, it comes to be minus the colloidal osmotic pressure and the capsular hydrostatic pressure. That comes to be 20 millimeters of mercury. Okay, capsular hydrostatic pressure. The capsular hydrostatic pressure is 20 millimeters of mercury. The blood colloidal osmotic pressure is 30 millimeters of mercury. The effective filtration pressure, it comes to 20 millimeters of mercury or 10 to 20 millimeters of mercury. The net opposing filtration pressure is blood colloidal osmotic pressure or capsular hydrostatic pressure. The addition of it, that is 50 millimeters of mercury. Okay. Now, when we talk about that the glomerular filtrate, it is same as plasma, but proteins are absent. Same as plasma, okay, but proteins are absent. But proteins are absent. Okay. So the glomerular filtrate is same as that of plasma, but proteins are absent. It contains mainly the non-colloidal substances. Okay. It contains water, ions, glucose, amino acids, water-soluble vitamins, okay, urea, uric acid. Now, so we have studied about the effective filtration pressure, the net filtration pressure, and NFP or EFP. Okay. Now, the glomerular filtrate, it passes through three layers, that is filtration slits. Three layers are there. First is filtration slit. Basement membrane, fenestrae. All the terms are already in the previous lectures we have discussed. Okay. Now see, the filtration slit is the innermost, which is having a size of 25 nanometers. Then in between we have basement membrane. Then outside we are having the fenestrae, that is endothelium wall of the blood capillaries in the Bowman's capsule. that is having small pores known as fenestrated epithelium. Okay. So now the plasma fluid that filters out from the glomerular capillaries into the Bowman's capsule of nephrons is called glomerular filtrate. It is a non-colloidal part. It possesses urea, water, glucose, amino acids, vitamins, fatty acids, uric acid, creatine, creatinine, toxins, salts, etc. Okay. The basement membrane allows passage of those substances which are less than 8 nanometers in size. Very important. Okay. Only allows those substances having size less than 8 nanometers. That is why the basement membrane is the most important part in filtration. Okay. Now, the glomerular filtration rate or GFR, that is around 125 ml per minute or 180 ml per day. Okay. The RBCs, WBCs, platelets, plasma proteins, they are colloidal part of the blood and do not filter out from the glomerulus. The glomerular filtrate is isotonic, very important, isotonic to blood plasma. It is isotonic to blood plasma. The glomerular filtrate or nephric filtrate it includes blood minus blood cells or plasma protein and blood minus RBC, WBC and platelets plus plasma protein, plasma minus protein, that is the glomerular filtrate. The GFR is the amount of the filtrate formed per minute in all the nephrons of the paired kidney. There is a sexual difference in male. The rate is 120 to 125 ml per minute. Okay. In female, it is 110 ml per minute. GFR is affected by volume of circulating blood, neural activity, stretch response to pressure of the wall of the artery. That is osmoreceptors. Okay. So around it is 125 ml per minute, or we can say 180 liters per day. The 180 liter of the filtrate is formed per day. Out of it, only 1.5 liter of urine is produced per day, which is 0.8% of the total filtrate. Renal plasma flow around 1250 ml, that is 25% of the cardiac output of the total blood, circulates through the kidneys each minute, and of this blood, around 670 ml is plasma. The later is called the RPF, that is renal plasma flow. The renal plasma flow is around 1250 ml of of total cardiac output. That is the 25% of the total cardiac output. Okay, the blood circulates through kidneys each minute, and of this blood, around 670 ml is the plasma. The later is called the renal plasma flow. RPF is 670 ml. The filtration reaction. This is the ratio of GFR to RPF and is called filtration fraction. It is known as what? Filtration fraction. Okay. So this is the ratio of GFR to RPF, that is renal plasma flow, and it is called filtration fraction. Very important. The filtration fraction. It is around GFR upon RPF. GFR, glomerular filtration rate, it is around 125 ml per minute. And renal plasma flow, it is around 670 ml. 670 ml. Okay. It rounds to come at 0.17. That is filtration fraction. GFR 120 upon 670, 0.17. Okay. Now, auto regulation of GFR, that is glomerular filtration rate, it's auto regulated. It is maintained by sending the feedback mechanism. Okay. Hence, the amount of GFR almost remains constant because of auto regulation. Okay. If the GFR increases, then the proximal convoluted tubule PCT cannot reabsorb ions. Cannot reabsorb ions. Okay. Renal blood flow is around 20 to 25% of cardiac output. Okay. The sensors, osmoreceptors, and chemoreceptors of macula densa helps to regulate GFR. Okay. The sensors of macula densa, osmoreceptors, they help to sense large amount of Na plus and Cl minus and water. That is why they help to regulate GFR by sensing Na plus, Cl minus, okay, water. It is basically maintained in three ways. One is the myogenic control or regulation. The afferent arteriole, it gets under the process of constriction to decrease the diameter. Afferent arteriole, which is bringing the blood, if it gets constricted, then less amount of filtrate will reach the glomeruli. Decreases the GFR. Okay. Then the tubuloglomerular regulation. It inhibits NO, which is a vasodilator, and causes more constriction. Okay. Then the sympathetic nervous system, it secretes norepinephrine, which causes constriction. Causes constriction, okay, which further decreases the GFR. So the GFR is auto-regulated by these three mechanisms. One is the myogenic regulation, second one is tubuloglomerular regulation, and third one is the sympathetic nervous system regulation, which secretes norepinephrine, which causes the constriction of the arterioles, and these arterioles further, decreases the GFR. Okay. Now, before coming to selective reabsorption, the renal blood flow, as we have studied, 20 to 25% of the total cardiac output. Okay. It is around 1100 to 1200 ml per minute. The renal plasma flow is 670. Filtration fraction is 0.17, that is 16 to 20%. Okay. If a person is suffering from diabetes mellitus, the glucose level in blood has raised the renal threshold, that is known as what, TM, or renal threshold, maximum threshold. Okay. The renal threshold is 180 milligrams per deciliter. Okay. So that is why if a person is diabetic, so if he goes for blood test, so the elevated sugar level in blood can be easily reflected, but the urine test goes false negative because it will only be, excreting glucose when the level crosses the threshold, that is 180 milligram per deciliter. Okay. The complete blood volume is filtered about 60 times per day. The complete blood volume is filtered by 60 times per day. Okay. Per day. Okay. The GFR is 180 liters per day. The GFR is 180 liters per day. Okay. Number of times the plasma is filtered is 60 times. It is 60 times per day. Okay. Now the selective reabsorption. The process of selective reabsorption, it includes basically, it is also known as the tubular reabsorption. Okay. It was discovered by Richard and the supporters. There are different parts of the nephron which basically have their affinity towards the threshold values of different substances. Okay. So when we talk about, so there are basically different categories for tubular absorption. The substances that are reabsorbed, some of them have high threshold stimulus substances, some of them are low threshold substances, and some of them are non-threshold substances. So I will be adding one slide so that I could, you know, add this information. So tubular reabsorption. So we have covered all the points regarding GFR, the glomerular filtration. Okay, now the tubular reabsorption, also known as the selective reabsorption. So there is a category of the substances which are known as what, high threshold substances. These substances are maximally reabsorbed. These substances are maximally reabsorbed. Example: water, glucose, amino acids, Na plus, Cl minus. Okay, these are high threshold substances. Then, what is renal threshold? Upper limit up to which a substance can be reabsorbed. It is the upper limit up to which a substance can be reabsorbed. So it is the upper limit up to which a substance can be reabsorbed, okay, by the nephric tubules, whether it is proximal convoluted tubule, whether it is the distal convoluted tubule, whether it is the loop of Hill. Okay, so the limit up to which a substance can be maximally reabsorbed, that the glomerular filtrate is having that substance in that amount. So if the tubules can taking the that nutrient from the glomerular filtrate, so it will make it or it will create its, the renal threshold. It is known as what? The renal threshold. Understood? Is this clear? Yeah. This renal threshold is different for different substances. Okay, like for example, glucoside, 180 milligram per deciliter. Low threshold substances. Okay, low threshold substances means that these substances, they have less tendency to get reabsorbed. Okay, up. The fact is that how your body will decide whether the substance is required at greater amount or lesser amount. Yeah, it can be decided by the body because it everything is dependent upon the feedback mechanism, right? It is basically dependent on the feedback mechanism. So whether it's required or not required, it is very important to understand that why it is necessary. Okay, it is necessary because if the thing is to be reabsorbed, it will become the part of the blood again, and a body can reutilize the substance. Okay, so less threshold substances which are having less, the less or the, you know, the required amount to get reabsorbed is low because the requirements are low. The body is less dependent on those substances. Okay, so these are the, what should I say, low threshold substances, partially reabsorbed. Example is urea, uric acid. Non threshold substances. These substances are not reabsorbed. For example, creatinine. So whenever if anyone goes for the, renal function test, anyone goes for the renal function test, RFT. And if the urine is having more amount of creatinine, then there is some type of renal failure or which is occurring in that person. The creatinine, hippuric acid, inulin, they all are the substances which don't have any threshold for getting reabsorbed. Okay, understood? Because of these processes of reabsorption, the urine can be concentrated up to 4 times than human blood. I mean, that is why our kidneys are metanephros, metanephric kidney, and we secrete a hypertonic urine because the kidney needs to conserve all these materials. Okay, these materials are required to be conserved because they can be used again. Okay, so this is what about I should say on the different types of threshold stimuli or the substances. Okay, like high threshold, low threshold, non-threshold. Okay. So moving to the next topic, I will be talking about the different regions of selective reabsorption. Okay, like PCT, proximal convoluted tubule, it is the pivotal site for reabsorption, main site for reabsorption. It is the main site for reabsorption. Okay, so it is the main site of reabsorption for different types of materials, especially the ones which are having high threshold, like glucose, amino acids, ions, which are reabsorbed by active transport, are reabsorbed by passive transport. Okay, there are some ions which are reabsorbed by active transport, there are some ions which are reabsorbed by passive transport. So, glucose, amino acids, passively transporting ions, actively transporting ions. Okay, active uptake of the ions reduces the concentration of the filtrate and an equivalent amount of water passes into the peritubular capillaries by osmosis. This is very important point. Very important point. To maintain the charge electrically neutral. Okay, so active uptake of ions reduces the concentration of the filtrate and an equivalent amount of water passes into the peritubular capillaries by osmosis. Here, 80% of water is reabsorbed by passive transport. By passive transport. Okay, so this is the proximal convoluted tubule. Okay, 60% of the glomerular filtrate is reabsorbed here. 60% of the glomerular filtrate is reabsorbed here. Okay. Water, Na plus, Cl minus, bicarbonate ions, glucose, vitamin C, amino acids, potassium ions, little amount of urea, uric acid. Okay. So the active uptake of the ions reduces the concentration of the filtrate and equivalent amount of water passes into the peritubular capillaries by osmosis. 80% of water is absorbed by passive transport. It is also known as obligatory water reabsorption. मतलब होना ही होना है, ठीक है ना? Obligatory water reabsorption. Most of the important buffers like bicarbonates is also reabsorbed from the filtrate. Proximal convoluted tubule absorb nearly 80 to 90% of the filtered bicarbonate. Some urea is reabsorbed by diffusion. The rest remain in the filtrate for to be removed in the urine. Okay. So the proximal convoluted tubule is actually making 60% of the reabsorption of all the nutrients in the glomerular filtrate. Okay. Mechanism of urine concentration of counter current mechanism. This is very important. Okay. I would like to make one diagram as well. Okay. Add one slide before this counter current. So if you want to, sketch out the that which region is basically, more favorable to reabsorb a particular substance. Okay. So PCT is all for this. So if I'll make a rough diagram, this is thin descending loop. This is U pin, or this is thin ascending loop. Then comes the thick ascending loop. This is just a rough diagram. Okay. So starting from this PCT, or proximal convoluted tubule, I'll mark it in black. Yeah. So till PCT, we can say the urinary filtrate is isotonic. Water is reabsorbed, Na plus is reabsorbed, K plus is reabsorbed, Cl minus is reabsorbed, amino acids are reabsorbed. All this region is peritubular capillaries. Okay. Ammonium ions, okay, in the form of ammonium bicarbonate. Bicarbonate ions are reabsorbed. Okay. H plus is given out in the luminal filtrate, in the lumen. Okay. Vitamin C, glucose. Vitamin C is reabsorbed, glucose is reabsorbed. Okay. So the osmolarity of urinary filtrate is 300 osmoles per liter. The moment it comes down. So here we can see the osmolarity is 400 osmoles per liter. Okay. It is basically permeable to water. It will give slow diffusion of water will take place. Water. Okay. This is the descending limb. Okay. The moment descending limb comes up with the continuity of thin descending limb, mean the thin descending limb is going to add and thin descending limb is going to add up. The osmolarity becomes 600. Further, it keep on removing the water, keep on losing the water, make it more concentrated. It becomes 800 to 1000 and at last it becomes 1200, which is a very hypertonic form. Okay. Now after this, the moment it reaches to the thick ascending limb, it reabsorbs Na plus, Cl minus, K plus, okay, Ca2 plus, Mg2 plus. This is ascending limb. Okay. At the level of DCT, at the level of DCT, it becomes hypotonic because now it will release all the metallic salts, solutes, and water will come in. Water will come in. Okay, so it becomes hypertonic. At the level of DCT, distal convoluted tubule, with the help of the ADH, aldosterone, RAS system, it will help to maintain the amount of water and the salts. Okay, H plus show an antagonistic movement with K plus and H4 plus. Okay, to maintain the pH, H plus they are basically given in the filtrate luminal side, Na plus. Okay, it further releases the H2O. Okay, and urea is maximally excreted in the collecting duct. Okay, urea, CO2 in the collecting duct region. I will make you write as well that which region is basically involved in the reabsorption of which part. Okay, and the urine is excreted. Okay, so this is just a rough picture that how the urine formation takes place, how things goes on from PCT to DCT and so on. Okay, now mammals form hypertonic urine. We have already understood in the previous slide. The urine is made hypertonic with the help of counter current multiplier system. Very important. Counter current multiplier system. What is this counter current multiplier system? It is basically a mechanism which will help in concentrating the urea by maintaining high concentrations of NaCl and urea in the medullary interstitium. In the medullary interstitium. So the urine is made hypertonic with the help of counter current multiplier system. This process takes place in the loop of Henle and vasa recta, and it mainly involves basically it involves the interstitial medullary interstitial regions like loop of Henle, vasa recta. Okay, it mainly involves. I will write here medullary interstitial region. Okay, loop of Henle and vasa recta. Okay, in PCT, urine is isotonic. The descending limb of the loop of Henle is permeable to water only. Only. Its surrounding tissue fluid is hypertonic, hence the water moves out and it becomes, as it moves in descending limb, by passive transport. Therefore, the filtrate in the descending limb becomes more and more hypertonic. Okay. So counter current mechanism basically it is to increase the amount of NaCl in urea, to increase the amount of NaCl and urea in interstitial, medullary interstitial regions, especially the loop of Henle and vasa recta. Okay. The loop of Henle forms countercurrent multiplier. The loop of Henle forms countercurrent multiplier by actively transporting Na plus and passively transporting urea. Okay. So the loop of Henle forms countercurrent multiplier. Multiplier. Okay. The countercurrent multiplier basically increase the active transportation of Na plus increases and passive transport of urea increases. Okay. The ascending limb of loop of Henle is impermeable to water, and that is why the solutes keep on moving. Okay. Now the water will move, but with active transport. So the descending limb, the water was moving with the help of passive transport, but the during ascending limb, the water will move through active transport. Hence, the filtrate becomes hypotonic. The water it further re-enters into the descending limb of the loop of Henle. The collecting duct always pass through the hypertonic tissue fluid. Hence, water comes out osmotically, making the filtrate hypertonic. In the urine which is passed out, okay, which that is hypertonic. In the collecting duct, glomerular filtrate is known as urine. The term urine first time used in collecting duct. Okay, collecting duct. Vasa recta is a very important region as it is mentioned Vasa recta and loop of Henle. Vasa recta is freely permeable to water. Very important. Vasa recta is freely permeable to water and urea. So Vasa recta sorry to water, NaCl and urea. It is freely permeable to water, NaCl and urea. Okay. Higher will be the intake of proteins, higher is the formation of urea. Hence, concentration of medullary interstitium will be more and more high, mean hypertonic, and the urine becomes more concentrated. Okay. Persons more on protein diet excretes more urine as it is hypertonic and concentrated. Okay. This is, you can see the filtrate entering the descending limb becomes progressively more concentrated as it loses water. The water is coming out 300 to 600, 900, 1200, hairpin loop. Okay. This is the peritubular region, the blood or the vasa recta. Blood in the vasa recta removes water, leaving the loop of Henle. This is vasa recta. Vasa recta is freely permeable to water, NaCl and urea. Okay. This is vasa recta. This region is vasa recta. Okay. Now, as the ascending limb, it becomes hypotonic from 900 to 600, then the 300 like that. The ascending limb pumps out Na plus, K plus, and Cl minus, and filtrate becomes hypoosmotic. Okay, it pumps out all the ions plus K plus Cl minus. Okay, and becomes hypoosmotic. So this is the counter current mechanism. Now we will discuss in the next turn about the DCT ascending limb, okay, and the hormonal control. Okay, what are the different hormones which will play an important role in controlling all these functions? Okay, till then, bye bye. Take care. Stay safe. Stay healthy.